We left off talking about bacteria and archaea, these simple single celled organisms. Okay, and we talked about photosynthesis evolving, changing the atmosphere of the world, which stopped new types of organisms forming. And so anything from here on out probably evolved from the same organism. So we can take our ancestry and trace it all back down to the same organism or group of organisms. So looking at eukaryotes, okay, and these are the cells we really focused on when we talked about cells way back in, I think, chapter 4. So eukaryotic cells have a composite ancestry, different components derived from different lineages. It's quite interesting when we really look into this. Uh, so we have archaea-like nuclear genes, so genes in our DNA that look at genetic processes. DNA replication, transcription, translation looks very similar to what we see in archaea. But when we look at metabolism and the formation of membrane within those genes, it's more like bacteria. And so we've got this weird composition where we share similarities with both of these organisms. Um, and the reason for this, or what is likely the reason for this, is that we see in bacteria and archaea, gene transfer is not uncommon, where genes from one organism will get picked up by another organism and then made part of its genome. And now it's picked up those genes from something that was completely different. And so that's likely why eukaryotes have these differences. So within eukaryotes, we've got our DNA, right, as all living things do, and it's all surrounded by a nucleus. Okay, this is something that our prokaryotes do not have. Okay, so this is one of those defining factors, pretty much the main defining factors within making something a eukaryote. So we see that some modern bacteria have internal membranes, these compartments, not necessarily surrounding their nucleus, their DNA, but we do see internal membranes. And so what we see is the way that we probably got this is through infoldings of the plasma membrane. Because if you remember when we talked about the plasma membrane, we called it a fluid mosaic. And so this membrane can move around and it can change its shape. And that's exactly what it's doing in this infolding. So these portions of the membrane fold in on itself and then pinch off. And we now have these internal membranes within the cell itself. And so the bacteria, archaea, whatever it happened to be, that made these internal infoldings, perhaps they had some sort of advantage. It helped them survive. When they surrounded their DNA, it protected it. It didn't have the same significant mutations that occurred in other organisms. And so they survived and produced more offspring. And so through this process of now natural selection in this infolding of the membrane, we now have internal membrane structures like the nuclear envelope, the endoplasmic reticulum, eventually the Golgi apparatus, and so on. And so this is likely how we got these internal membranes. And so these are bacteria that currently exist. And we see this infolding of this membrane. All of these lines in here are membrane that has folded in into the interior of this bacteria. Here with this Gemata obscuriglobus, it does actually have a double membrane, as we would see in a nuclear envelope, surrounding the DNA. So should we consider this one a bacteria or not? It's up for debate, but it has what we essentially would call a nucleus, which is very interesting. Okay. Looking at mitochondria and chloroplasts, okay, these organelles that process energy. So in all eukaryotes, we've got mitochondria, which does cellular respiration. And then in photosynthetic organisms, we've got chloroplasts, which are doing photosynthesis. So the way that these came into eukaryotes is what we call the endosymbionte hypothesis. So they're essentially bacteria that were engulfed into an early eukaryotic cell. And then basically they created a symbiosis. They created a relationship where the bacteria processed energy for the eukaryote. And in return, that bacteria was protected and it got the building blocks and things that it needed to process energy. Over evolutionary time, these things became intertwined and connected to one another and no longer could exist separately. And so what we see is that mitochondria, their genes look very similar to a bacteria. And then in chloroplasts, those organelles, the DNA that exists there are really similar to cyanobacteria. So we see that mitochondria and chloroplasts have their very own DNA, completely separate from the DNA of the eukaryotic organism itself. So here's rickettsia, very similar to mitochondria. Okay. Other pieces of evidence for endosymbiosis. Okay. We see that bacterium infects a eukaryotic cell. We can get this endosymbiosis, and it can happen multiple times. Okay. They'll become incapable of living independently from one another, and so they more or less become the same organism. Okay. And so here we see a photosynthetic organelle, very similar to a cyanobacterium. Okay. Same thing here, where this chloroplast is very similar to a cyanobacteria. This video is going to do a great job really animating this process and discussing where this mitochondria, the chloroplasts, they came from and why we see them so much in eukaryotes. And it's going to give some other evidence of organisms that we see where this has happened multiple times, where we've seen this endosymbiosis. Okay? And so that's the end of chapter 18, the origins of life. Again, this is a field of study that's underway, and a lot of new things are being learned all of the time.